बिस्मिल्लाहिर्रहमानिर्रहीम। आज हमने ज़ूम में जो कुछ पढ़ा था ये वीडियो उसकी समरी है ताकि आप समझ जाएं। आज हमने अपने न्यू चैप्टर पर स्टार्ट लिया था, चैप्टर 17 बायोटेक्नोलॉजी। फर्स्ट उसमें हमने ये स्टडी किया था दैट व्हाट इज़ बायोटेक्नोलॉजी। बायोटेक्नोलॉजी में किसी माइक्रो ऑर्गेनिज्म को लाइक बैक्टीरिया, यीस्ट वगैरह को यूज़ करके ह्यूमैनिटी के लिए कोई बेनिफिशियल प्रोडक्ट बनाया जाए तो उसको कहते हैं बायोटेक्नोलॉजी। Biotechnology is the use of microorganism for the welfare of humanity. Next, importance of biotechnology. Biotechnology is not a new field. It is an old field and it is related with other fields of sciences like fermentation, genetic engineering, tissue culturing. What is fermentation? Fermentation is anaerobic respiration. What is genetic engineering? Genetic engineering is the engineering of new organism by DNA alteration. Tissue culturing is the technique in which fragments of plants are cultured and grown in a lab. Fermentation is a type of cellular respiration. As already studied that fermentation is an anaerobic respiration. It occurs during the absence of oxygen, and during this process, the incomplete oxidation of food molecule occurs, and a little amount of energy is released. There are two types of anaerobic respiration. First one, alcoholic fermentation by yeast cell, and second one, lactic acid fermentation by bacteria. These two types are named according to their end product. Alcoholic fermentation usually completes in two steps. Number one, glycolysis. Number two, alcohol formation. Glycolysis is the breakdown of glucose molecule into pyruvic acid, and alcoholic formation from pyruvic acid, alcohol or ethanol is formed as shown in the figure 17.2. Lactic acid fermentation. And this process also completes in two steps. Number one, glycolysis, and second one, lactic acid formation. Glycolysis is same discussed above that it is the breakdown of glucose molecule and pyruvic acid is formed. And lactic acid formation, from pyruvic acid, lactic acid is formed as shown in figure 17.2. Next, yogurt formation. How yogurt is formed from milk. Skimmed milk which contain excess amount of fats. This is introduced in a culture medium where certain specific bacteria are present. This specific bacteria usually ferment the sugar, lactose, which is present in milk. Lactose is converted into lactic acid, and then lactic acid is converted into curd, and then they form yogurt. Fermentation products. There are different fermentation products as shown in the slide. Alcohol, yogurt, bread, cheese, pickles, soy sauce, chocolate, chemical products. Alcohol is usually formed when glucose molecule is broken down into carbon dioxide and alcohol is formed. Yogurt is formed from the fermentation of milk. Is already studied in above topic. Bread. Wheat dough is fermented to make bread. When yeast is added to the wheat dough, this is fermented to make bread. Cheese is formed when bacteria converts the lactose into lactic acid, then milk is converted into cheese. Pickles is usually formed from fruits and vegetables. Pickle is usually formed from the fermentation of fruits and vegetables. Soy sauce, it is usually formed by the fermentation of soybeans. Chocolate, it is formed by the fermentation of cacao beans. Chemical products, these are formed through fermentation process. Genetic engineering and its uses. Genetic engineering is the engineering of organism by DNA alteration as studied in the previous topic. As already studied that biotechnology is also related to other fields of sciences like genetic engineering. Genetic engineering has importance in DNA alteration, artificial manipulation, and transfer of genetic material. DNA alteration is the change in the DNA sequence. Artificial manipulation is the growth growth of organism by artificial means. Recombinant DNA technology, there are two organisms. One is the donor organism and the second one is the recipient organism. From donor organism, the DNA is taken and introduced into the recipient organism. So now this organism is called the GMO, genetically modified organism. And what is recombinant DNA? The DNA which contain the gene of both sources like donor and recipient. ये आज का हमारा लेक्चर था जो आज हमने पढ़ा था, ठीक है, ये उसकी समरी थी. थैंक यू सो मच.